So I'm Ben Twining, and I'm excited to lead this session with Steve Archer, who's also going to talk. And I'm excited that we're going to talk about microzooplankton this afternoon, so woohoo! So it seemed like it was about time to have a session on microzooplankton. I think that they can get a little bit overlooked sometimes when we think about carbon cycling and upper ocean processes and oceanic processes in general. And one reason I think, as we were talking before the session, is that they're messy, they're complicated. They don't fit into clean size groups often. They don't fit into clean physiology groups. And they're also really hard to sample. And those things alone make them sort of maybe make some of us want to not think about them. But they're important. And I think we're going to, today will be a good reminder of that. I think another reason that that they tend to get overlooked sometimes is that the sort of explosion of molecular tools that are now being applied to the ocean, I think maybe for simplicity, have begun to be applied at the bottom of the food chain, at the sort of bacteria and phytoplankton level. And I think that emphasis on the bottom of the food chain sort of lets us forget a little bit about the larger microbes, the protists. I think the other reason is because the emphasis on carbon export and carbon cycling of recent years leads us to think, when we think grazers, we think fecal pellets, we think carbon export from mesozooplankton, and that might also let us to sort of not remember quite so clearly how important the microzooplankton are to carbon cycling. But that ends today. So today we have 4 great talks on 4 topics that hopefully will explain or expand the diversity of the roles that they can play in ecosystems. So we're going to start with Diane Stoecker, who's going to talk about polyunsaturated aldehydes and their impact on microzooplankton grazing, growth and mortality. That's going to be followed by a talk by Steve Archer, who's going to talk about a new approach to measuring grazing rates in natural systems. And for anyone who has had to do a dilution experiment and knows how finicky those are, I think that's a tool that could be very useful to our community. Ben Ward is going to talk about mixotrophy and the importance of that, and Stephanie sort of set that up and set the table for that a bit this morning, which is great. And finally, Virginia Edgecombe is going to talk about protists in the deep sea, and an area that maybe we don't think about the grazing impact as much, and also talk about the symbiotic relationships that they can be engaged in. So hopefully we'll get a nice broad span of subjects, and we'll stimulate a good discussion afterwards. And with that, Diane, come on down. Let's see, do I need to...
Thank you, Ben.
Take care of that. Go for it.
Well, I'm going to talk about polyunsaturated aldehydes and their impact on microzooplankton grazing, growth, and mortality due to predation. And this research I'm going to talk about is really from 2 projects. One that I got involved in, which was a Mesocosm study in Norway. And my job as part of that study was to study microzooplankton grazing using the dilution technique. But I was worried that it wasn't working in that situation, and I was right, and I'll show you. And also a recent NSF project that we're still working on, and this is with Peter Larienta, Gaia Franza, who's sitting right there and knows everything, and Jamie Pearson, who works on Copepod and worked on this project with us. So, what are PUAs? Well, they're wound-activated oxylipins, and they're only actually produced when the phytoplankton cell is wounded or stressed or dying. So nutrient stress at the end of a bloom can cause this, mechanical stress, things like filtering, and also things like grazing can trigger the production of the oxylipins. And they're self-inhibitory, and this has been studied quite a bit. Like particularly some of the diatoms that produce them, if they're exposed then to the PUAs, they actually undergo a death cascade and release more of the oxylipin. And it's been hypothesized that they deter copepod grazing because In some situations, ingestion of them, the copepods will grow and lay eggs and look happy, but the eggs won't hatch because they can be mitotic inhibitors. But a few people, including Flynn and Rogin, have suggested that there really can't work evolutionarily as being designed to prevent copepod grazing because the timescales are so different. A copepod takes weeks to develop. A microzooplankton or a phytoplankton, you're talking hours to days. And also, microzooplankton are the major grazers, even on diatoms in the oceans. And I'm going to concentrate on one particular one. Ooh, sorry. Skeleton myrinoid, which a lot of work has been done on. Here's a little picture of it, full chain-forming diatom. And it produces two PUAs, octadienol and heptadienol in like a 1:10 ratio. And it also may produce other oxylipins and other compounds that we don't really assay or know about. So it's very complicated. And this is the Mesocosm study that we did during March back in 2012. And we were trying to get a Phaeocystis bloom, this line here, but instead we got a skeletonema bloom. And so you work with what you get. And we found very high PUAs total during the peak of the skeletonema bloom. And now I'm going to show you 3 grazing experiments that I did during that bloom. So how I got into this is I'd worked before for 3 years in the Bering Sea. And we did about 60 grazing experiments. But in a lot of them, the grazing wasn't significant. And in some of them, we actually got statistically significant negative grazing rates, which is sort of impossible. Okay, but it is possible if the growth rate of the phytoplankton is lower in the diluted treatment than in the whole seawater. And so from doing those experiments and some physiological measurements, I suspected that somehow dilution water produced during blooms could become toxic to the phytoplankton. And so in these experiments, in the mesocosm, I did a standard dilution experiment, except I added a treatment. And that treatment was taking the dilution water and passing it through a carbon cellulose filter, which removes organics, including the PUA. And our results. Well, if we looked at the rise of the bloom, that peak right here, and we looked at the growth rate, net growth rate of phytoplankton in the whole seawater and in the diluted treatment, there wasn't much difference where you would expect it because you're relieving grazing to be higher in the diluted treatment. But if we put that dilution water before we set it up through the carbon cellulose filter, all of a sudden we got increased growth. We did it also at the peak of the bloom, right here, and we didn't get any positive growth in the whole seawater, the diluted treatment, but in the carbon cellulose treatment we did. And when we did it here in the decline of the bloom where everything was just crashing and it looked like dirty dishwater, it didn't make much difference. Everything was just dying. So my conclusion from that is that during some types of dense blooms, preparation of filter with seawater can result in release of PUAs and probably other material that's inhibitory to phytoplankton. And when you think about it, it sometimes takes 2 hours to filter the 20 plus liters you need. And so you're stressing the cells, and also PUA, it isn't like it's a little bag in the cell. It's actually synthesized when the cell is injured. And so it's a process. So if you're sitting there for 2 hours dying, you have time to produce PUAs. Next, I'm going to talk about more recent experiments. This is in Chesapeake Bay in the coastal Atlantic. And these were with Peter and Jamie and Gaia. And we did dilution experiments to look at microzooplankton grazing and phytoplankton growth. Then we also did grazing experiments with copepods to see how PUA affected their grazing on phytoplankton and also microzooplankton. And we did this with natural assemblages at 5 experiments over the last 2 years, 3 of them in the estuary and 2 of them on the coastal Atlantic. And the design of these experiments are a little confusing. I'll try to go through it. But one treatment that we did besides the control was addition of dissolved PUA in the 1:10 ratio of the OD to HD. And we added it at concentrations that mimic what might be a light bloom, a moderate bloom, or a very, very heavy bloom of a Skeletonema marinoid strain that was very toxic, and made the assumption that maybe 10% of the cells would lyse and release their PUA. So these are the treatments we came up, like 2.2, 5.5, and 22 nanomoles total PUA. And we added them at the beginning of the 24-hour incubations, but PUA does break down. So we really don't know what the PUA concentrations through the whole experiment were. The other thing we did was addition of a high PUA strain of Skeletonema marinoid. And we also had a control strain that didn't produce PUA. And we added between 2,000 and 4,000 cells per ml for that treatment. And so that's comparable to a light to moderate Skeletonema balloon. Well, what did we find? Okay, here, these experiments 1 through 5. And first we looked at the growth rate of phytoplankton, and the black is the control, and the gray is the PUA treatment. And we really didn't find any difference in the growth rate of phytoplankton in these experiments between the control and the PUA addition. But when we looked at the grazing rate of microzooplankton, and 3 out of the 5 experiments, we saw significant decreases in the grazing rate of microzooplankton. So, how am I doing here? Okay. Effects of PUA on microzooplankton grazing depended on what they were grazing. Actually, when we broke it down, on Synechococcus, in some cases, the grazing rate on Synechococcus by microzooplankton actually increased. There was not much effect on small eukaryotes, but on larger eukaryotes, bigger than 5 microns, this would include the diatoms. In 4 of the 5 experiments, we saw a decrease in microzooplankton grazing on them. Well, what are the effects on the microzooplankton themselves? And we looked at the effect on dinoflagellates and also on ciliates. And basically, we saw inhibition of both dinoflagellate growth over 24 hours and ciliate growth in the medium to high PUA concentrations. But it was very variable and partly depended on the species. One species, Mesodinium rubrum, which is a photosynthetic ciliate, appeared to be less sensitive, and only the highest PUA caused a decrease in it. Something we've done recently with Hoshu Zhang in the Ocean Engineering Department here and with Matt Johnson is, does it actually affect the swimming of the microzooplankton? And this we did with Protoparidinium, a heterotrophic dinoflagellate. And what we found was at T0, you get a nice sort of normal distribution. The cells are helically swimming. But after exposure, even for a short time to PUA, you get some very small, some cells that swim very slow and some that swim very fast. So there's a real change in swimming behavior. And I'm not going to show it here, but Hoshu just recently sent me on Friday the data for a tintinid. And we see in that species 2 effects on swimming. So why is that important? Well, swimming can affect your ability to find food and graze, and it can also affect your susceptibility to copepod predation. So here, we did some experiments. They're not all analyzed yet. What happens if you actually add this high PUA skeletonema? And in this experiment, we saw declines in the dinoflagellate and in the ciliates over 24 hours. So their growth was depressed with the PUA. Well, what effect does it have on copepods and copepod grazing? We didn't really look what it did to copepods, but what it does to their grazing. If we look at chlorophyll, there is some increase in grazing on chlorophyll, okay? Not much, but some in 4 out of the 5 experiments. If we look at ciliates, in 4 out of the 5 experiments, it really pretty dramatically increased grazing on ciliates. And clearance rates for ciliates are almost always higher than for chlorophyll anyway, so the bars are bigger. And if we looked at grazing by copepods, and this is like all with Acartia, on dinoflagellates, in 2 of the experiments we saw an increase in grazing rate on dinoflagellates. So this should cause a trophic cascade, where you're actually increasing, by increasing grazing pressure on the microzooplankton that do most of the grazing in the oceans, you may be relieving phytoplankton from grazing pressure. We did some experiments that were similar with adding the PUA-producing skeletonema. And this is a summary of 5 experiments. And what we see is the red is the ciliates. That's the clearance rate of the copepods for the ciliates in the control, and it's much higher with the added skeletonema, even though there's more phytoplankton food available. And it looks like there might be an effect also on the dinos and the chlorophyll, but that wasn't statistically significant across the 5 experiments. So, summary so far. Well, they do produce these inhibitory metabolites that can be reduced in producing dilution water. So this can result in underestimation of grazing. I don't think it's always a problem, but I think if you're working in bloom situations, it's something you should think about. There is a big effect on microzooplankton and microzooplankton grazing. It's in general depressed, particularly on the bigger phytoplankton cells. And that PUA actually stimulates copepod clearance of most types of prey. So this is our sort of conceptual model that actually Gaia drafted this. Here we have phytoplankton, and grazing by microzooplankton here on phytoplankton is inhibited, particularly on the bigger ones, even though grazing on the really small things like Mesocosm can be enhanced. But grazing by copepods on the microzooplankton is stimulated. And it can also stimulate grazing by copepods on some phytoplankton. But the net effect of this is that it can relieve big phytoplankton from grazing pressure. So what are the consequences of all this for the, I'm going to have plenty of time for questions. The consequences of this for the food web is that it disrupts the growth grazing equilibrium, and that really provides an opportunity for further bloom development. And not every diatom bloom produces PUA, and even diatom blooms of Skeletonema marinoi don't always produce PUAs. There are PUA-producing strains and non-PUA-producing strains. But there are certain places where this seems to be particularly important. And one is like in the Barents Sea off Norway, because we took Skeletonema water from the Barents Sea in our Mesocosm and then had this big bloom. And also in the Adriatic Sea, this is very important. Our experiments were done in Chesapeake Bay and coastal Atlantic. And we looked at the background PUA levels there, and they were either undetectable or very low. And I think one real question that we hope to address is, well, what happens if you do similar experiments, but in the Adriatic, where the plankton is used to seeing Skeletonema marinoi and PUA. And Peter and Jamie have been going over and doing some experiments in the Adriatic, so hopefully we'll have an answer to that. A lot of the grazing problems I saw in my experiments in the Bering Sea were mixed blooms of Phaeocystis and different diatoms. And I was focused on Phaeocystis at the time, but I think there's a real potential that this type of phenomenon is very important in Arctic waters. Thank you.
Questions for Diane?
Thank you, Diane. It's very interesting that you are providing an explanation for negative grazing rates, which I think everybody who's done delusion experiments has observed. I don't quite... so you're saying the PUAs would hurt the grazers. You show that it can hurt the micrograzers. But in the highest dilutions, you're reducing or decoupling grazing altogether. Wouldn't it be then actually encouraging that and accentuating grazing rates and not
well, the thing is,
inhibiting
in a
them?
dilution experiment, you're really measuring grazing by how much faster the phytoplankton grow when the grazers are diluted away, right? So you expect a higher net growth rate of phytoplankton, let's say in the 10% whole seawater than the whole seawater.
The idea is that the growth rate is the same. It's just that the grazing rate is reduced.
Well, the net growth rate.
The net growth, right.
Yeah,
the
which
net
is the only
growth
thing you can
would
really
be enhanced.
measure.
Yeah.
So if that's true, that's what you expect. But then if the PUA is inhibiting the growth of the phytoplankton, so it isn't higher, or it might even be lower than in the whole seawater, that's when you get problems.
So the key
Even
is
if
that
it has no effect on the grazers.
right. So the key is that it would hurt the phytoplankton themselves, not the grazers.
Right, and that's a little different than what we saw in these experiments here, because in the stuff in the Chesapeake Bay we didn't see much effect on phytoplankton. But in those experiments in the Mesocosm, we did, and the only thing I can think of, we were working in a mesocosm at very high concentrations, and I don't really know how much PUA was in that dilution water that we took out. It may have, like in these experiments, we used 5 nanomolar. I don't know what we used in a dilution experiment, because you don't measure the PUA in the water. And I might add, the PUA analysis is not easy. It isn't like you can just do it and get a number. It's expensive, and you get the number months later.
Thanks.
So incubations obviously are used in a lot of assays, primary production, nitrogen fixation, nutrient uptake. Do you think PUAs might actually have, what do you think about PUAs possibly having an application in terms of determining whether or not the organisms in your bottle are stressed, maybe as an indicator of like bottle effects or something like that?
I think that would, unless the techniques for determining PUA are a lot better than they are now and cheaper, no. One thing we did do I didn't show them here, but the initial experiments in the Bering Sea, in about 14 of those, Joachim Goes, who does variable fluorescence work with me, and we looked at the variable fluorescence of the phytoplankton in the whole seawater and then in the diluted treatments. And what we found was in many of those experiments, the ratio of variable fluorescence to fluorescence maximum was lower in the diluted water. which means they're stressed, okay, as an indicator of stress. And that also correlated with what the microzooplankton grazing rates were. So you measured, not that the microzooplankton grazing rates were lower, but you measured a lower rate usually when you got this depression in variable fluorescence in the diluted treatments. So that's one thing you might use is variable fluorescence.
I'm going to ask a question while you're... Diane, trying to get a sense of how broadly this could apply, like what kinds of systems this applies to. It seems like these are fairly high biomass areas where you get the PUA to accumulate. And do you have a sense of how variable the PUA production is across phytoplankton strains as well? I mean, is this skeletonema really unique in its production?
You know, in the Adriatic is where they've done the most work, okay? And they've isolated a lot of different strains of skeletonema that vary in their amount of PUA production. And actually the one we used didn't match the PUA that the highest ones are supposed to produce. It was like around 2 phenomoles per cell, where the highest one is around 10. So there's this whole spectrum between 0 and 10. And we know it's important in the Adriatic. We know it's important up around Bergen in the Barents Sea, but there aren't very many measurements of PUA. It's also produced by Phaeocystis, and that's how I originally got interested in it. But our Mesocosm experiment, we only got Phaeocystis at the end, and the PUA didn't go up. So apparently some Phaeocystis maybe can produce PUAs and some can't. So, and that's a really interesting question. Why should it be important in the Adriatic and not in the Chesapeake Bay?
Yeah.
I think you just answered my question. I was just wondering if you had any thoughts, Diane, on what causes one species to produce it in one location but not in another.
That's a mystery to me. And also I think there are other things that don't produce PUAs but produce something similar. Because we've done experiments with when Leptocylindricus was blooming, and we tested it for PUA and it doesn't produce it. And Ponart has tested cultures for it and it doesn't produce it. Yet every time we try to do a grazing experiment when there's a Leptocylindricus bloom, we get these, you know, negative rates and strange stuff. So, you know, so I think there are a lot of mysteries out there.
Great. All right, let's thank Diane again.